very good day to all. I am going to take the very first lesson of this academic year in biozoology, reproduction in organisms. Reproduction in organisms. So here in this lesson we are going to see whatever the topics which we are going to cover is modes of reproduction, and then the next one is asexual reproduction, sexual reproduction. So these are the three agendas of this particular lesson. So here what we see in this, the first one is that the modes of reproduction. Modes of reproduction in this, what are the types and other things we are going to see in that. And what is the agenda of reproduction? What is the agenda of reproduction? So here in this reproduction, when I say reproduction, is nothing but the biological process in which the organisms produce their own kind of young ones. It's a biological process. When I say it's a biological process, that says it is applicable to the living things. When it is applicable to the living things, the organisms they produce their own kind of young ones. So that is the agenda. And then what happens to the young ones after reproduction? They grow and mature to repeat the process, and they were resulting in the continuation of the species. So when I say the agenda of reproduction, it is nothing but the continuation of species. Continuation of species is more essential to perpetuate the organisms in this earth, and introduces variations. Variations in that every time when an animal is undergoing evolution, it has to show some sort of variations. So that variations that may be in the genetic constitutions also. So here we are able to get the variations in the organisms which are essential for the adaptation and evolution of their own kind. So the agenda, when I say agenda, this agenda refers to this particular word, essential for adaptation and evolution of their own kind. Okay. There are two types of reproduction. Two types of reproduction. Basically, you know, right from your lower classes, it is well known for you. Anyway, I have to make you recall certain things. Asexual reproduction and sexual reproduction. Asexual reproduction. So here, what happens? When I say A, so here this is the absence of sexual gametes, and the reproduction is done in the absence of sexual gametes. And sexual reproduction, it is enhanced only with the help of the sexual gametes. So here in asexual reproduction, you will see this does not involve the fusion of gametes or change in the number of chromosomes. Does not involve the fusion of gametes. Then what happens? What happens if there is no gametes? What will happen? Obviously, the body itself will undergo division, amitotically or mitotically. So that is one. And another one, the offsprings that arise by this asexual reproduction from a single parent. Here there is no biparental union, only a single parent undergoes division. And another one, this or from a multicellular organism inherit the genes of their parent. The same parent, whatever the genetic constitutions they had, has to be replicated, or it has to be just like that, a set or bunch of chromosomes has to be forwarded to the next generation or to the daughter cells. So that is the main agenda for this. That is asexual reproduction. And when I say sexual reproduction, so here what happens? This requires two organisms. Asexual, I said only one organism is necessary, whereas in sexual we need two organisms. Here each organism has a specialized cells called the gametes. So we call these cells of the specialized cells because they are involved in the reproduction. They are responsible for the variation, for the continuation of species, also for the evolution. Isn't it? So that is why we call it as a specialized cells called the gametes. And here the animals have both male and female gametes. That is, the male cells have sperm and the female one is a gamete. And suppose if it is a plant, you have the pollen. The pollen is called as the sperm, and the egg is called as the ovum. So let us not go deep into that. And sexual reproduction, this happens when two sex cells, they fuse together. They have to fuse together. Only after that, the fusion or the syngamy, that is responsible for the creation of a genetically different organism. Okay. Asexual reproduction. So where does it occur? Where does it occur? Asexual reproduction occurs mainly in the lower organisms or in the kingdom Protista, bacteria, archaea, and some of the multicellular organisms with simple level of organization. When I say that there is simple level of organization here, they doesn't have a definite organ or organ systems arranged in the body. So that is why we call them as a simple level of organizations, and different modes of asexual reproductions are present here. One is the fission, second one is the sporulation, and third one budding, gemmular formation and fragmentation, finally the regeneration. So here we are going to discuss about these 6 topics in this lesson, and the first one we are going to open is the fission. Fission, when I say fission, you might have heard in your lower classes. Fission, when we say fission is the division, it has to divide into maybe 2 cells or more than, or multicellular cells, whatever it be, but the fission refers to the division. So it is a division of the parent cell into 2 or more identical daughter individuals. It has to resemble exactly the copy of the parent cell. The daughter cells has to resemble exactly the copy of the parent cell. And there are 4 types of fission we see in this. One is binary fission, multiple fission, sporulation and strobulation. 4 types: binary, multiple, sporulation and strobulation. So the first one is a binary fission. So binary fission. So here children, you have to see this, the term, the very term refers you, or that itself gives you the explanation. Bi means 2. Fission, we say that it divides into 2, binary fission. The parent organism divides into 2 halves from daughter cells. It divides into 2 halves from to form the daughter cells. Okay, that's point number 1. Next one, what says this is followed by the karyokinesis. Later on it is carried over by the cytokinesis. What will happen here is that when I say karyokinesis, it is the division of nucleus. Always in a cell, the nucleus divides first. Nucleus divides first, and then it is divided by mitotically or amitotically. Have you heard of this word mitotically or amitotically? When I say amitosis, that means this is also similar to mitosis, but one difference is that this doesn't form the chromosomal apparatus like the spindle fibers or chromosomal apparatus, all those things. That is the main difference you have between mitosis and amitosis. And then the division of cytoplasm is followed later on. So we call the term karyokinesis for nuclear division and cytokinesis for cytoplasmic division. And the resulting offspring is genetically identical. So this is what we want from the binary fission. So this should be genetically identical. And the binary fission, here also we have some subdivisions among what do you say about 4 divisions are there in binary fission. That is depending on the plane of division, which plane or the axis, the direction from where it starts, where it going to end, So depending on the plane of division, binary fission is of 4 types. One is simple irregular binary fission, transverse binary fission, longitudinal binary fission, oblique binary fission. So the first one, simple irregular binary fission, simple irregular. Children, you see that it is a very simple and binary fission which is very irregular. It is called as irregular. The reason is that you can see the amoeba. In amoeba what happens? There is no definite shape. There is no definite shape for amoeba. That is why the plane of division is hard to observe. That is the reason they have given the word. It is very hard to observe from which plane or which direction it is going to start. It is unknown. And the contractile vacuole. See, contractile vacuole, there are parts which are present inside the amoeba, or the contractile vacuole, nucleoli, cytoplasm, all these are present inside. So first what ceases to function and disappears is that the contractile vacuole, it ceases. It ceases means it doesn't want to do its function anymore. So it disappears. After that the nucleoli disintegrates, then only the nucleus divides mitotically. You should know the difference between the nucleus and the nucleoli. You should know what is contractile vacuole. What is the purpose of contractile vacuole? All these you might have learnt in your lower classes itself, but never mind. The contractile vacuole, which is actually it is performing the excretory function. So here it ceases its function and disappears, nucleoli disintegrates, nucleus divides mitotically, the cell then constricts in the middle. So, whatever it is, it constricts in the middle and then the cytoplasm gets equally scattered. So, whenever it gets constricted, the cytoplasm has to be equally scattered and divides into two daughter cells. You can see in the diagram. See here the diagram, irregular simple binary fission, contractile vacuole here, nucleus and the hyaline area, it is all present inside. So, the nucleoli disappears, contractile vacuole disappears, after that the constriction appears in the middle and it deepens, dividing the parent cell into two, finally getting the distribution of cytoplasm and all the parent material, forming the daughter amoebae. The next one is the longitudinal binary fission. Here also you will understand that when I say longitudinal, the axis or the plane of division starts longitudinally, that refers to that. Here the nucleus and the cytoplasm divides in longitudinal axis of the organism, longitudinal axis of the organisms in flagellates. So, in flagellates this type of fission only in the daughter cells. In flagellates what you might see, if it has a flagella for moving, for locomotion, so then you call it as flagellates. Here this type of division we will find only one daughter cell retains the flagella. One daughter cell retains the flagella, whereas the basal granule divides into two and the new basal granule forms a flagellum into the other daughter cell. Example for that is verticilla. See in this diagram, you can see that the flagellum is present here. This flagellum, it is the parent cell. When it divides into two, the flagellum comes to one daughter cell alone, Then the other daughter cell will be provided with the other flagellum by the presence of basal granules which is present at the base of the flagellum. Now, the basal granules divides into two. After dividing, the basal granule substitutes one flagellum for the newly divided cell. So, at the end of division, both the parent or the parent, from the parent, the daughter cells, they get equal daughter flagella and nucleus, everything will be shared for both the daughter cells. So, from the parent you can see this in this diagram, you can verify with this diagram. The next one is transverse binary fission. Transverse, so here also the plane of division is in the transverse axis of the individual. Paramecium and the planaria is the example which is given here. In this one, interesting factor is that the paramecium, it has got both macronucleus and the micronucleus. Two types of nucleus are present. The macronucleus divides by amitosis and the micronucleus divides by mitosis. So, here you can see the presence of both the nucleus, macronucleus and the micronucleus in paramecium. This is an interesting factor which you see in the paramecium. So, here the macronucleus divides by amitosis and then the micronucleus divides by mitosis. Finally, you can see two daughter cells formed with the equal number of cells which are provided in the parent cells. Okay. Then the next one is the oblique binary fission. So, when I say oblique, here also the plane of division is oblique. So, this is seen in the dinoflagellates. Dinoflagellates are nothing but it is also a type of algae. So, here the example given here is the Serraceum. See Serraceum here, this undergoes oblique binary fission. And the next one is multiple fission. Multiple fission. So far we have seen binary fission, which is irregular, and then transverse, longitudinal, oblique. Now we are going to see about the multiple fission. In multiple fission, what happens? The parent body divides into many similar daughter cells simultaneously, immediately, one after the other. So, it divides simultaneously. The many similar daughter cells were produced simultaneously from the parent body. And then what happens here also, as it is. See here, one difference is that first the nucleus divides repeatedly without a division of the cytoplasm. First the nucleus divides into number of cells, innumerable number of cells or divisions. After that, later the cytoplasm divides as many parts as that of nuclei. Remember this, as many parts as that of nuclei. Which one? The cytoplasm. And now this encircles one daughter nucleus, resulting in the formation of many smaller individuals from a single parent. many smaller individuals from a single parent. So, here first nucleus divides repeatedly without division, without the division of cytoplasm, and random. Finally, at the end only you can see the cytoplasm divides into as many parts, encircles one daughter nucleus, resulting in the formation of many smaller individuals from a single parent. This is one type. And another one is repeated fission. That is another type of fission we will see, that is the repeated. Now, here what is the difference between these two is that in the multiple fission, one of the type is repeated fission. Here it produces 4 or more individuals, equal cells and the cell division. The young ones, what happens to the young ones is that they do not separate, until the process is completed. They do not separate. They will be present within the parent itself, within the body of the parent itself. Then finally, after complete division, after complete process is completed, the ones do not separate until the process is complete. That is what we see here as a difference we will see in verticilla. The next one, in multiple fission, one example we have taken in Plasmodium for reference. So, here in Plasmodium, have you seen Plasmodium? Have you heard of this word Plasmodium? Plasmodium is nothing but the malarial parasite, you would have learnt in your lower classes. So, Plasmodium in multiple fission, it occurs in the zyzone stage. Here the zyzone stage is there. See, one of the stage in the Plasmodium life cycle is the zyzone stage, and those cells are called that is the zygogony. Here what happens, resulting in the daughter cells, and they form a type of cell, what you call it as merozoites are formed as a result of this zygogony process, and the cells are called as zyzone. This is one type of multiple fission we observe in Plasmodium. And another one is the oocyte stage. When I say oocyte stage, here you might be understanding what is the term oocyte. Here in sporogony, we call it as sporogony. The resulting daughter individuals are called sporozoites. So, multiple fission we have taken example, one in Plasmodium forming the merozoites in this process, another one in the oocyte stage. Here sporogony cells are formed, that is at the end of this daughter individuals formed are called as sporozoites. So, multiple fission in Plasmodium we will find 2 types of cells, one is merozoites, another one is the sporozoites. Then another type of multiple fission, where and all you find this case we have taken as an example. In amoeba also we will find this type of fission, multiple fission. But here it is Nsystem, we call it as Nsystem. What do you mean by this word Nsystem is that it is covered by thick layered cyst wall, a thick layered cyst wall, and then the inner content protects the future young amoebae. So, that is what we see here, that during unfavorable condition, that it is not favorable for them to exist, that is the decrease in temperature, scarcity of the food, there is no food for them, and a decrease in temperature, scarcity of the food. What happens to that is that the amoeba withdraws its pseudopodia. Pseudopodia means the false legs, it is withdrawn, and now this secretes hard chitinous cyst wall. This is the hard chitinous cyst wall, 3-layered cyst wall you can see, and becomes inactive. What happens? It goes inside and it is getting inactive, and this forms an Nsystem. This process having 3-layered cyst wall, nucleus, cytoplasm, all these together, and inside the daughter nucleus of present, we call it as Nsystem. It is getting inactivated during unfavorable condition. Okay. Now, when favorable conditions are written, what happens? The encysted amoeba divides by multiple fission. After that, what happens? It divides by multiple fission and produces minute amoebae. As many number of daughter nuclei are produced, and they started producing minute amoebae. What do you call it as? Pseudopodiospores or amoebulae. Pseudopodiospores. So, pseudopodiospores means the spores here with the false leg. Once again, the amoeba is the young ones are produced. What do you call it as? Amoebulae. Okay. The cyst wall. Now, what happens to the cyst wall? Absorbs water. What is the function of the cyst wall hereafter? It has to absorb water and breaks off. It has to breaks off, liberating the young pseudopodiospores. So, young pseudopodiospores are liberated, and each one with a false legs. That's what it has to move, isn't it? For locomotion it needs a leg, false leg. What do you call it as pseudopodia? Now they start feed and then they grow rapidly to lead an independent life. So the agenda is fulfilled. The function of the cyst wall, it absorbs water, breaks off and then liberating the young pseudopodia spores with each one with the false legs, that is the pseudopodia. They started feeding, they grow rapidly to lead an independent life. The next topic is strobulation. This is a special type of transverse fission called strobulation. Occurs in some of the metazoans. It's coming between protozoans, poriferans, we can see like that, metazoans. Here the transverse fission occurs simultaneously giving rise to number of individuals. As many number of individuals are produced which do not separate. They do not separate. You see here, they do not separate from each other. So this type of case we find in Aurelia. The example here you can see in the picture, Aurelia aurita. And there is another type of division, what you call it as plasmotomy. Plasmotomy, the division of multinucleated parent. The parent itself is a multinucleated parent. Now it is divided into as many individuals of daughter individuals, multinucleated daughter individuals with the division of the nuclei. The parent is a multinucleated parent and multinucleate daughter individuals are produced with the division of nuclei. As many number of nuclei are divided. So here the nuclear division occurs later to maintain the normal number of nuclei. Here what they have to maintain is that the number of nuclei has to be maintained for the normal number of nuclei. So that is the agenda. See here the division of multinucleated parent into many multinucleated daughter individuals with the division of nuclei. Nuclear division occurs later to maintain normal number of nuclei. If there is any variation, it is maintained at the later stage. That you find in Opalina and Pilomixa, that is the giant amoeba. Now next one, the sporulation. Sporulation, here we will find an amoeba. See here, sporulation occurs in amoeba. In amoeba it multiplies by sporulation without encystment. Previously we have seen encystment of amoeba. Now the sporulation occurs, that means in the adverse condition it has to undergo an inactive stage, an inactive condition. Then you call it as sporulation. Here there is no encystment, without encystment, and the nucleus breaks into several fragments, small fragments. See the nucleus divides into as many number of small fragments or chromatin blocks. Here we call it as chromatin blocks. That term is used here, and each fragment develops a nuclear membrane. Later on it develops a nuclear membrane around that. See the spore here with the nuclear membrane surrounded by the cytoplasm. One single one, you can see the nuclear membrane, cytoplasm content, and the spore case, that hard case you call it as a spore case. During favorable condition, what happens? The parent body disintegrates and the spores are liberated, each hatching into young amoebae. The next stage, budding. The very term explains you budding. Buds are produced from the parent body. They produces one or more buds, each grows into a young one. So here this you will observe in Hydra. So what will happen? They separate from the parent to lead a normal life. That is number one. And there are two types of budding. One is exogenous, another one is endogenous. In sponges, have you heard of sponges? Poriferans. The buds constrict and detach from the parent body and they develop into a new sponge. And if it is an exogenous, endogenous means within the body, exogenous means outside the body, the buds are formed on the outer surface of the parent body, that you will find in Hydra. You can see the picture, two types of budding, exogenous and endogenous. In the Leucosolinia, that is a type of sponge. You can see the bud developing from outside the body, that is the bud develops. Later on, after complete formation, gets detached from the parent body. And in Hydra also you can find the formation of the bud, the growth you can see here, it deepens, after that gets cut off and it leads a independent life. The next one, in Hydra what happens is that when food is plenty, when it has plenty of food, the ectodermal cells, that means the ectodermal cells means The layers, the first layer, which is the outermost layer, increases to form a elevation. First a small elevation is formed, and then the endoderm and ectoderm are pushed out to form a bud which contains the interior lumen in continuation with the parent's gastrovascular cavity, and then the bud enlarges and develops mouth and a circle of oral tentacles at its free end. When fully grown, the bud constricts at the base and finally separates from the parent body to lead an independent life. See here the Hydra, which is having the outermost layer, ectoderm, and then endoderm inside. In between you have Mesoglia. It deepens and forms a lumen. This is a gastrovascular cavity. The green color what you see is the gastrovascular cavity, and then at the oral end it forms the free tentacles and a basal disc which is attached to the base. Endogenous budding. Endogenous, within the body itself the budding develops. So that's what endogenous. Seen in Noctiluca, the buds are formed inside the cytoplasm. So here also we can see the number of buds are formed inside the cytoplasm, and many remain within the body of the parent itself. Within the body of the parent itself. Next, gemmules. The next one we call it as gemmules. So the internal buds are formed by asexual reproduction in some of the fresh and marine sponges. Here we call it as gemmules because it is a hard ball with an internal mass of food laden archaeocytes. See, in gemmules and sponges you will find this type of case, that is the gemmules, which is having spicules on the outer side covering the body like the skeleton. It is giving a support for that, and internally you have the archaeocytes, food laden archaeocytes are present. So this structure is the internal bud formed in the asexual reproduction of the fresh and marine sponges. And the next one is during unfavorable condition, what will happen? The sponges, they disintegrate. The spicules and the outer covering, everything gets disintegrates, and the gemmules can withstand adverse condition. Adverse condition due to that the presence of food laden archaeocytes, whatever is present within the body of the gemmules. Later, when favorable conditions return, the gemmules begin to hatch. See, the archaeocytes, all these are present here. You can find this is structure. Then the pinocytes, colonocytes, all these are the internal structures which are present here. And archaeocytes are nothing but the food laden particles. Then and there you can see the spicules are present, giving a support to the gemmule. Fragmentation, the last one, fragmentation. Here we say the body breaks into fragments, and each of the fragment has a potential to develop into a new individual. That is, each fragment has a potential to develop into a new individual. In sea anemone, you can have a sea anemone as an example. Here pedal laceration is seen. Pedal means the leg, the base of the disc. Laceration is seen here. Here, that is, the lobes are constricted off from the pedal disc. Each one, when it is completely developed, it is lacerated from the parent body. It is taken out from that. Each lobe grows mesenteries. So here, this grows mesenteries as well as the tentacles to form a new individuals. When I say mesenteries here, it is the internal part. See here, the oral tentacles are present, the body here, and the base, the pedal laceration. See here, you can see in this picture, the mesenteries are developing to give food for the developing young ones. Then you will see a type, what do you call it as Epolysis. Epolysis in Tinea solium. So here Epolysis means, what is Epolysis? The ripe or the gravid proglottids. See, in tapeworm you can see each one, you call it as a proglottids here. See, at the lower side of the organisms, as it grows on, matures, you can see the proglottids which are present, the segments, we call it as the mature proglottids, and later on the matured one, they are all shed off from the body. What do you call it as the gravid proglottids? So here the posterior end of the strobila, each one is called as a strobila. The ripe one or the gravid proglottids that are at the older end or seen at the posterior of the end of the strobila, which are regularly cut off either singly or in a group. That's what you call it as Epolysis. So the term is, what is Epolysis? Regularly they are cut off from the body. What do you call it as Epolysis? And this is done mainly to transfer the embryo. The agenda of this Epolysis process is to transfer the embryo from the primary host to the secondary host. Now, in this tapeworm has got two hosts, one is the primary host, the another one is the secondary host. Primary and secondary, man and the pig are said to be the primary and the secondary host. For that, to transfer the embryos, they have to shed off the strobila, and that process you call it as Epolysis. And the regeneration process, regeneration, regrowth. So, nothing but the regrowth in the injured region, where there is an injury in the body part. So, there the growth occurs. That regrowth or the injured region of the injured region, we call it as regeneration. There are two types here, one is morphollaxis and another one is epimorphosis. Morphollaxis and epimorphosis. So, the first one is morphollaxis. Here the whole body grows from a small fragment. The whole body grows from a small fragment. So, that you will observe in Hydra and the planarians. So, here the fragments usually retain a polarity. They retain a polarity. Suppose when you have a fragment, doesn't mean that that fragment will grow either upside down or in a straight form, having the anterior and posterior axis, all this. So, that is what we call it as retain a polarity, as the head and the tail region. That region is maintained here, retain a polarity. When cut into several pieces accidentally to produce the oral ends with the tentacles and aboral ends with the basal disc. Oral end is nothing but the, what is that, to feed. That part we call it as the oral end. So, with the tentacles, and the aboral end we call it as the basal disc are present here. So, they maintain a polarity. You can see the regeneration in Hydra. See Hydra, it is, even if you cut it into as many fragments, each one maintains the polarity. See, each and every bit it produces one complete form. Produces the oral tentacles and the basal disc is formed. Even in planaria also, if it is cut into three parts like this, here the head will produce both the lower part of the organisms, and the middle piece will produce the complete head and the tail, and the lower part of the tail will produce the entire anterior region. So, this sort of regeneration you can observe in the Hydra and planaria. Another type of regeneration is epimorphosis. So, what happens here is that replacement of last body part only, the last body part. That means here also you have two types in that. One is reparative and another one is restorative. Reparative and restorative. Here in reparative generation, only certain damaged tissues can be generated. Only certain damaged tissue. See, in starfish, only the part, one of the arm is damaged here, only that tissue is replaced here. Whereas restorative, you are restorating, that means here the severed body parts can develop. See the tail of the lizard. So, this is observed in the lizard, the tail of wall lizard. If the body parts are severed, it happens only in the tail. We have observed mostly in the restorative regeneration or epimorphosis condition. We can find this wall lizard as an example. And for reparative regeneration, you will find starfish as the example. Thank you for observing.